नमस्कार दोस्तों, welcome to channel Toxicology on Board. Today in this video we'll discuss about question number 23. The topic will be related to the LD50 value and briefly we'll discuss about dose response curve. So these are the important guidelines: OECD 420 guideline, OECD 423 as well as OECD 425. These three guidelines are the one which deals with the acute toxicology studies. Let's see what question we have. What modification of LD50 assay is described by the following? Okay, what is the description given in the question? We'll see. Animals are dosed one at a time, starting at an estimated dose LD50 dose. If the first animal survives, the next one receives a higher dose. If the first animal dies, the next one receives a lower dose. The spacing of the dosing generally is adjusted by a factor of 3.2. Important thing you have to remember, the factor 3.2. So let's see what options are given. Option A, the up and down method. Option B, approximate lethal dose method. Option C, acute toxic class method. Option D, fixed dose procedure. The right answer is option A, the up and down method. As you can see, these questions are taken from this American Board of Toxicology. What is the explanation and difference given? In the up and down method, the animals are monitored for overt signs of toxicity until death. Method A, method B, and D are all modification of LD50 assay. Up and down method, approximate lethal dose method, as well as fixed dose procedure, designed to reduce animal numbers and or in increase quality and quantity of information collected. So reducing the animal numbers, again this is nothing but application of 3R principle. Okay, let's see what we can learn from this question, the content which are related to this particular question. Briefly understand what is relative toxicity and traditional LD50. Dose that is expected to be lethal to 50% of the treated subject. Here, experimental animals. And LD50 is a unique to each compound. So LD50 is specific to every chemical and is statistically calculated based on available data. The error is dependent on the number of animals used and the inter-animal variation in response. So whenever this statistical method you are applying, we'll discuss about the errors. Okay. Since an LD50 is a calculated value that varies with the quality and quantity of the data, it is always accompanied by an estimated of associated error, such as probability range and confidence limit. Limits of probability range are arbitrarily selected by the experiment. Okay, experimenter. The more data that are used to determine the shape of the curve, the more accurate the value becomes. So more and more data points you collect during that experiment, more accurate LD50 value you'll get. The statistical procedures that are commonly involved in toxicology are the same procedures that are used in other biological sciences. So Wilcoxon method is there, some Litchcoxon method or something like that. Some statistical methods are there when you derive this LD50. Toxicologists therefore need to understand that statistical procedures are most suitable for specific data sets being evaluated in a given study. Okay, this is a typical dose response curve. You can see NOEL, LOEL can be and also LD50. So let's see in next slides how this LD50 are related with respect to, we learn with respect to one dose response curve. So LD50 is obtained from the curve by drawing a horizontal line from the 50% mortality. So on the y-axis you will have a response, on the x-axis you will have dose. Okay, curve by drawing a horizontal line from 50% mortality point on the y-axis to where it intersects on the curve. So this is the place where it intersects. Okay, at the point of intersection here a vertical line is drawn and this line intersect with the x-axis at the LD50 point. So this will be the LD50 for this particular chemical A. Okay, It is evident from the curve that information with respect to the lethal dose for any percent of animals, okay, 95% of the animals or 5% of the animals may be derived by similar procedure. So here 50% as response, extra move it towards the dose response curve, and from that intersection point move towards the y-axis, you will get 50%. Similarly, if you want a 95%, you can select this particular point, move towards the dose response curve, extra down towards y-axis. This particular point will be considered LD95. Similarly, here by at 5% response, you can move towards the curve and extrapolate down to y-axis. From that intersection point, you'll get LD5. That is what he's saying. The lethal dose for 84% of the animals represent +1 standard deviation from the LD50, and lethal dose for 16% of the animals represent -1 standard deviation from the LD50. So from this LD50 graph, you can get LD95, LD5, LD84, LD16. Okay. Next, this is again very important. This is a table which is given in the case of uh, in from the textbook. I forgot to give the reference. Probably it might be Clayson or Hayes. You just go back and check. If you can see, these are approximate LD50 of selected variety of chemicals. Okay. So the range of LD50 for different toxicants can vary significantly. If the LD50 for one compound for is a few micrograms, as you can see in the case of botulinum toxin, okay, here the LD50 units in mg per kg, it is 0. Total 5 zeros, 1 mg per kg, it will come under micrograms, like probably maybe 0.001 microgram. That is what is saying. For one chemical, for example, botulinum toxin, LD50 might be in micrograms compared with several grams. Here you can see in case of alcohol, LD50 is 10,000. If you divide it by 1000, you will get 10 grams. So several grams for another compound, the difference between the two compound is highly significant. Okay. So numerical wise, you can see for certain chemicals it might be in micrograms and certain chemicals in terms of grams. Okay. If it is possible, try to read these all these chemicals. What are the target organ toxicity? There might be some historical incidents, some significant toxic effects will be there with respect to these things. Try to learn. For example, tuberculin and arrow poison. You can learn. You can learn about strychnine sulfate, which is derived from strychnine nux vomica. You can learn about recreational drugs like morphine and derivatives. Okay. You can also learn about ethyl alcohol mechanism and toxicity pathways, metabolism. You can learn about botulinum toxin. You can learn about dioxin, how it was used during World War II, tetrodotoxin, bufotoxin, which are there in the marine species. So like this, whenever picrotoxin also, which is a dye, okay, I believe. So DDT related history is there. So what I'm trying to say is, once this table comes, try to read all these chemicals related toxicity. This topic also you try to cover. Okay. So next we'll see chemicals that are produced death in microgram doses are commonly thought as extremely toxic. So in the previous table, botulinum toxic is extremely toxic. Ethyl alcohol less toxic. So so extremely toxics are LD50 with 1 mg per kg or less. Okay. Other chemicals may be relatively harmless following doses exceeding several grams. Right? Sodium chloride also it was. Toxicologists have devised categories based on amount of each chemical necessary to produce adverse outcomes. So here one classification is given. Similarly, you will have EU classification is there, GHS classification is a classification labeling package is there. If you are a toxicologist working on risk assessment, you know that ECA classification is also there. For each chemical based on LD50, it can be categorized the nature of the toxicity, and this type of data is used in case of SDS authoring. Okay. An example of such categorization along with respective lethal doses is given below. Okay. So next we'll try to understand what is this particular graph. In this example, the LD50 of compound C is less than LD50 of compound D. Similarly, in the case of efficacy graphs, dose response curves, you will understand the potency, the therapeutic index. Similarly, two different chemicals C and D, their LD50, their dose response curve, how that we will learn in this particular graph and example. LD50 of compound C is less than LD50 of compound D. How we will see? The reverse is true for LD5. For example, 50 here it is, no? Here this is the dose which takes this is the dose for chemical C to cause 50% mortality. If you look at this one, chemical D, in order to produce 50% mortality, the dose will be here. So LD50 of compound C is less than that of compound D. So LD50 is more for compound C is more toxic than D. But coming to LD5, if you see for chemical C, the LD5 is this value. Okay, and for chemical D, the LD value is LD5 value is this. So that is what is trying to say. The reverse is true for LD5 for the two compounds. If the low if the low dose is the only consideration, compound C will be classified as being less toxic than compound D because the compound C has higher LD5. Okay, then compound D. However, when the comparison is made between LD50 LD50s, compound C is more toxic than compound D. If you are comparing LD5s, LDC is the compound D because compound C will be classified being less toxic. Okay, C is less toxic than D. LD50 C is more toxic than D. So what do you understand? What is the take home message from this is that it is therefore apparent that both the slope and shape of the dose response curve. This is important. Both slope of the curve. That's why we'll say is the dose response curve steep curve. STEEP steep. They will say you'll come across this word whenever you're doing any 28-day toxicology study. You come across this term dose response curve. Steep is the steep curve. How your dose response curve is it linear? Or it is the non-linear. So such kind of terminologies you will come across whenever you are dealing with the graphs. So it is therefore apparent that both the slope and the shape of the dose response curve may be most significant when comparing the relative toxicities of two or more compounds. Right? In this case, C and D are compared here. So let's see safety versus toxicity. So again, in this graph, we'll try to understand from these two, from this dose response curve A as well as B, chemical A and chemical B, how they are behaving. If the two drugs produce quantitatively identical effect, for example, you have acetaminophen and you are having aspirin, both are producing the same identical effect. The drug that produces the effect at the lower doses. So, whenever you're developing a drug, for example you're developing an antihypertensive drug. Drug A is showing the same efficacy as 50 mg and drug B is showing the efficacy at 75 mg. So what do you, as a researcher, as a scientist, or a pharmacologist, which drug would you like to bring it into the market, or the regulators will give approval? Definitely drug A. See, same effect you can get it from the lowest dose, then it is more beneficial because you are not subjecting the patient to the higher dose. Unnecessary exposure to another chemical is not needed. Same thing you can achieve with another drug with a lower dose. Definitely regulatory will give approval to that particular dose. Okay, so that is the reason you mean to say the drug that produces the effect with lower dose is said to be more potent of the two drugs. Okay, so nowadays, if you look into pharmaceutical manufacturing also, there is a something term called HPAPI, highly potent active pharmaceutical ingredients, because the advancement, the technology is moving towards that type of drugs into the market where the same efficacy can be achieved at a lower dose. Okay, the margin of safety is the dosage range between the dose producing a lethal effect and the dose producing the desired therapeutic effect. So this is the margin of safety, the difference between the two dose response curve. One the efficacy curve, another one is mortality curve. Okay. If the slopes of the dose response curve for the two medicines are parallel, okay, then the margin of safety between the two medicines may not be different. MOS will be, I think it will be 1, I believe. You go back and check. This margin of safety, also known as therapeutic index, is obtained experimentally. The figure 2.4 shows a hypothetical dose response curve for a chemical agent administered to a uniform population of animals. You have to always select your experimental group of animals. Okay, this is hypothetical dose response curve. Curve A represents therapeutic effect of the chemical and therapeutically effective dose for 50% of the animal is calculated from the curve A, right? Curve B represents this is nothing but toxic effect, lethality. LD50 is calculated from curve B. The margin of safety is represented by LD50 by ED50. LD50 by ED50. This is what. How you are getting LD50 means 50% extrapolate this intersection point, move towards y-axis. This is your LD50 dose. Okay, and this will be your effective dose ED50. As shown in figure 2.4, if the curve B lethal effect is shift towards the left, if you're moving this curve towards this direction and approaches curve A, the margin of safety would be decreased, MOS. So if MOS is less means it becomes very challenging for the physician or the clinician to tighten the dose in the case of patients. For example, the is digitoxin. Digitoxin is the drug which is having lowest margin of safety. That's why digitoxin should be given to the patient only in the presence of that one reason, not only that is the only reason, in the presence of a well-trained physician. Okay, anesthetic drugs also similarly in the case. Chemical induced adverse effect may be either reversible or reversible. Let us understand the term irreversible tissue damage, irreversible death. Okay, this is another dose response curve. You can go back and read in detail, pause the video and go through. Each of these effects, including absence of any effect, can be expressed in the form of dose response curve. Once you have a data, put it, you can get it in the form of graph. Okay. The overall picture of safety and toxicity of any chemical dose related can be graphically demonstrated. That is what I was saying. Each effect can be represented by a curve with a specific slope and range of doses. Okay. The data are derived from approximately conducted experimental studies are then subjected to statistical analysis that produce acceptable virtual numbers, effect dose A, B, C, or 50% of treated subjects. The virtual numbers are simply a concise form for expression of the experimental results. It should not be recognized as a being applicable only to a specific initial experiment. So in this video we have learned about initially there was a question about LD50 and the answer was up and down method. So in the first slide only I've kept about three different guidelines: OECD 420, 423, 425. Before that there used to be some another guideline called OECD 401, which is absolute, which is no longer there in the OECD series of test guidelines list. That has been replaced with 420, 423 and 425. 420 is fixed dose method, 423 is acute toxic class method, and 425 is up and down procedure method. And our answer was up and down procedure. How you will get these things is these are the decision trees which are given at the end of the OECD guidelines. So we'll make another video on only on this decision trees. As of now, you can pause and you can go through and understand this guidelines because length of the video is already 70 more than 15 minutes. um, Thank you. Be curious. For any questions, please comment and you can write a mail to arya.priyanka18 at the gmail.com. Like, subscribe, and follow my YouTube channel, Toxicology on the Board. Uh, Thank you. All the best for the exam.